My name is Dr. Tirtha Doniger, and I work in the Bioinformatics unit at Bar-Ilan University. I will be guiding you through the practice sessions of Lecture 4. In this hands-on demonstration, we will be using Galaxy to illustrate the analysis of DNA sequencing data. Galaxy is a free, web-based platform for data-intensive biomedical research. Its goal is to develop and maintain a system that enables researchers with little or no informatics expertise to perform difficult computational analyses. Galaxy is freely available for use at the link provided below. Okay, let's begin. Click on the Login or Register link located at the top of your Galaxy interface. Click on Register, unless of course you already have an account. This process will only take a few moments of your time, but it will allow you to save and share your data in between sessions. Once you have completed the registration process, you may now log in. As a registered user of the Galaxy main server, you are now entitled to store up to 250 gigabytes of data. Once you are logged in, you will note in the upper right-hand corner a small bar which will always show you how much of your allocated resources you are currently using. If you exceed your disk quota, no further jobs will be run until you have permanently deleted some of your data sets. The right-hand side of the screen contains the History panel. When data is uploaded or an analysis is performed on existing data using Galaxy, each output from those steps generates a data set. These data sets and the output data sets from subsequent analyses are all stored by Galaxy in the History panel. The left side of the screen contains a list of programs that are available to perform analyses. We will begin by retrieving the data for today's session. In the left-hand panel, click on the Get Data link, followed by Upload File. Then click Paste Fetch Data and enter the URL into the box that appears. You can leave the type as Auto Detect and simply set the genome to the HG19 genome. That's the human genome version 19. And then click on Start to begin retrieving the data. You may now close this window. Depending on the size of the data, it can take several minutes to several hours to complete. Our data set comes from the whole genome sequencing of the subject known as NA12878, a human, female, a Utah Mormon with Northern and Western European ancestry. This data is from the 1000 Genome Project and was generated on an Illumina HiSeq sequencer. For demonstration purposes, the input file, the FASTQ file, contains only partial data, those reads mapping to a subsection of chromosome 10. In addition, the original data was paired end, and we will only use the single end for the purpose of this demonstration. The whole raw dataset is available at the 1000 Genome website. Once the upload status turns green, it means the upload is complete. You should now be able to see the file in the Galaxy History panel on the right. The dataset will have a very long name, as it's named after the full URL we retrieved it from. Optionally, once the file is in your history, click on the pencil icon to the right of the file name. Then select the name box, and you may change the name to anything you wish, or just remove the entire URL and keep the file name. Then click Save, and the file name will have been edited. To view the file, click on the icon that looks like an eye. This file contains our input reads. It has a .fq, which is a FASTQ extension. Let us begin to understand what a FASTQ file looks like. These files are the standard output of the Illumina sequencer. A FASTQ file contains 4 lines per sequence entry. The first line begins with an @ character and is followed by a sequence identifier and an optional description. Line 2 is the raw sequence data. Line 3 begins with a plus character and is optionally followed by the same sequence identifier found in line 1, although not always, as in the case here. And line 4 encodes the quality per base for the sequence in line 2 and must contain the same number of symbols as letters in the sequence. The quality value represents the probability that the corresponding base call is incorrect. This value is encoded in ASCII text in order to save storage space on computers. There are 3 different options for FASTQ file formats in Galaxy. One is FASTQ Illumina, FASTQ Sanger, and FASTQ Solexa. They each differ in the way the qualities are encoded. Older data from Illumina may be in FASTQ Solexa format. Newer data from Illumina is generally in FASTQ Illumina, and data downloaded from Geo will generally be in FASTQ Sanger format. However, Galaxy usually is able to guess, based on the data, what quality encoding was used for your data. Once we have obtained the data, the first step is to verify the quality of the data. In the left-hand panel, click on or under the FASTQ Quality Control. It's already open. And we can now click on the FastQC Read Quality Reports. Here you will choose the file which is already chosen from our history of the FASTQ file that we are now interested in analyzing. Once we have chosen the correct file, we can press Execute. Okay, you'll now note on the right-hand side in the history that these are currently running. You see a clock next to them. When they have completed, they too will turn green. The FastQC program aims to provide a simple way to do some quality control checks on raw sequence data coming from high-throughput sequencing pipelines. It provides a modular set of analyses which you can use to give a quick impression of whether your data has any problems of which you should be aware of before doing any further analyses. As you can see, they have turned green in our history, so now we can begin by looking. They have it as a FastQC on Data 4 as a web page. Click on the eye, and this would be the output file. Let's take a detailed look at the FastQC report. On the left is a summary of the quality control categories that are evaluated by the FastQC program. A green check indicates the data passed these QC. A red X or an orange exclamation mark would indicate cause for concern. So let's begin looking at each category. We have the basic statistics, which includes a file name, what file type, the encoding, it detected the encoding of the file, how many sequences overall. There are 230,282 sequences. If any of the sequences had been flagged by the Illumina machine as poor quality, that would be indicated here, and followed by the sequence length, how long are each read. Here, each read is 148 bases. Now, if there was a range or variety of lengths, it would be indicated here with the range from the smallest to the largest, but because they only indicate one length, that means that all the sequences are the same length in this file. And the %GC means the overall GC content of the file. The next category is the per base sequence quality. This view shows an overview of the range of quality values across all bases at each position in the FASTQ file. Now, for each position in the read, a box whisker type plot is drawn. The elements of the plot are as follows. The red line that you see is the median value in that position. The yellow box represents the interquartile range between 25 to 75%, and the upper and lower whiskers represent the 10% and 90% points. The blue line that's snaking through across all the positions represents the mean quality at each position. Now, the y-axis shows the quality scores. The higher the score, the better the base call. The background of the graph divides the y-axis into Very good quality scores, that would be in the green area. Calls of reasonable quality, that would be in the orange area. And calls of poor quality in the red area. Now, just so you know, in general, almost all the sequencing technologies, the chemistry degrades with increasing read length. Thus, it is quite often that we see that the quality of the reads will degrade as the run progresses. So we may see base calls falling into the orange or red area toward the end of the read. And in order, you can trim off that part if the quality does degrade below a reasonable level. Generally, the acceptable rule of thumb is that if the read is above 20, then that is considered an acceptable quality for the read. The next category would be the per tile sequence quality. Now, for each read, we know their XY coordinate on the tile, on the Illumina chips that they're actually sequenced on. And occasionally, you'll have an area of the chip or the tile that has bad, poor imaging or some kind of other problem. And this allows us to see if there's any area on the tile that perhaps, because of where the sequence was sequenced from, might have caused a problem. As you can see here, everything looks, the quality in this tile looks good all over. The next category is the per sequence quality score. Here we're looking at the overall quality score over the entire sequence. And this just gives us an idea if there's a subset of sequences that have a low overall quality value. It could be because they were poorly imaged, like they were maybe on the edge of the tile or things like that. But we should see that if there is such a group of reads that have an overall low quality, that it's really the minority, and that most of the reads, as you can see here, have a high overall quality of 36, 37, and that is the mean sequence quality score. Next up, we're looking at the per base sequence content. This looks at each position in the read and what's the proportion of each base at that position. Now here you see an overall flat line in each position, which indicates that the proportion is overall evenly distributed, or really the distribution reflects the genomic base pair content of the genome. But here, for example, I'll show you an example where we would see sequencing reads that still contain a primer. And in this case, you note that the unusual distribution of the base pair content at the beginning of the read indicates that there's still primer sequence attached to the sequence. In this case, before alignment, we would need to trim the sequence in order to be able to align them. And hopefully after trimming, we would see a similar distribution to the sequences that we have in our current library. Okay, following that, we have the per sequence GC content. Again, this is measuring the GC content across the whole length of each sequence. And it compares it to a modeled GC theoretical distribution. Now, in a random library, you would expect to see a normal distribution such that the peak would correspond to the GC content of the underlying genome. If you would notice, let's say, an unusual shape distribution, that could indicate maybe there's a contamination in your library, or maybe there's some kind of biased subset of sequences that are causing an unusual distribution. Next, we have the per base N content. So if a sequencer is unable to determine with sufficient confidence what base it is, it'll replace that base with an N. And so we would hope that there are few or no N's in our library. As we can see here, it looks across all positions for N's and finds 0% N's across our library. Next, we have the sequence length distribution. Illumina sequencers generate uniform sequence lengths, but other sequencers can generate wildly varying lengths. And even within libraries that are uniform lengths, there are pipelines that will trim poor quality bases. So you may get a distribution of sequence lengths. Here, all the sequences are 148 bases. Following, we have the sequence duplication levels. This is to give us an idea of how diverse our library is. And in a properly diverse library, as you see in this case, most of the sequences will be toward the left of the graph. In the case of over-enrichment or contaminations, you might see peaks coming on the right-hand side of the graph. Next, FastQC looks for overrepresented sequences. In our case, there are no overrepresented sequences. Now, generally, in a normal high-throughput library, which contains a diverse set of sequences, generally there are no individual sequences that are overrepresented. An overrepresented sequence could indicate either some highly biologically significant finding, or it may indicate that your library is not as diverse as you expected, or there's some kind of contamination. Again, important information to know. Finally, using a list of commonly used adapters, FastQC checks to make sure that your library is free of adapter content. Generally, after FastQC, if the reads were found to contain some kind of primer sequence or low quality bases, we may first need to trim or remove some of the sequences. But once we've established that the sequence quality of the library is adequate, we may now proceed to the next stage, aligning the sequences to the genome, which we will visit in our next segment.